Hello students, welcome to Learner's Planet. Now today again we are continuing with the class of biology, and the topic to be discussed is fundamental unit of life. So next, another process by which the molecules can be transported, the molecules can move across the membrane is osmosis. Which molecules? Water molecules. Now osmosis is a phenomenon in which the movement of water molecules occur through a selectively permeable membrane from a region of its high concentration to a region of its lower concentration. Now as we are saying that this movement is occurring, so if it is moving from a region of higher concentration to a region of lower concentration, then definitely the direction in which the molecules are moving around, it will depend upon the concentration of solute also in the surrounding medium. Because you can think of this point now, water is not a plain medium, it is not a completely aqueous medium. They are having certain molecules or the solutes dissolved in it. So how many or what is the concentration of this solute in the water will actually change the original concentration of the water. For example, if there is a solution in which there is, let us say, 40% sugar, that means what? 40 grams of sugar is dissolved in the 100 ml of water. In the another case, let us assume 10 gram sugar is dissolved in the 100 ml of water. And you can imagine now this particular condition. What will be the content level of water in each situation? The one which is 40% will have less number of water molecules as compared to the 10% solution over there. So in the 10% solution, the water molecule will be higher. So the amount of water which will cross in the cell or anywhere in the medium will actually depend upon this. It will be more in case of 10% solution, osmotic rate, whereas the osmotic rate in the 40% solution is going to be comparatively less, because already the water molecule has been already utilized to dissolve the sugar over there. So number of water molecule is less over there. So definitely the concentration of solute is going to affect the osmotic rate in the particular system. So we can now imagine that how this process is taking place. Actually, osmosis, we are saying it occurs through a selectively permeable region. So if we consider a plain water, a normal distilled water, then how this process is going to take place? So let us take this example. There is a beaker. Now this beaker has been partitioned by placing a semi-permeable membrane in between. On this side, the dilute solution, or we can say there is more water on side A, whereas on side B there is comparatively less amount of water. So in between, this semi-permeable membrane is present. Now what will happen? These water molecules, they tend to move across the membrane. But in which direction? A to B or B to A? It will be from direction from A to B side. Why? Because towards A side, the water molecule is high. The level of water is high in the case of section A, and in the B it is low. Now can you imagine another situation? Let us suppose there are two regions. Each is having equal amount of water. The level, we are saying the level. Fine. Now A side, it is concentrated. Fine. That means it is 40%. 40% solution of any compound you can assume. Whereas the level is same towards B, but it is dilute. So what do you imagine? If there is 40% sugar and here is 10% sugar, then where the water molecule is going to be high? Definitely in the case of 10% solution, as it is dilute. Oppositely, it will be the water molecule will be low towards side A. So now what will be the direction? It will move from B side to A side. And of course the process is taking place through a semi-permeable membrane. So this is how we can understand the process of osmosis. First case, we have simply taken a water, but the level of water was different. So in such condition, it will move from a high concentration region to a low concentration region. But what if they are now mixed with the solute? So it will depend the site where the solute is high. That will definitely have the low amount of water. So it will move towards, the water from outside will move towards the lower side. So this is one of the example, or it is just an illustration to explain the process of osmosis. On the basis of concentration of the solute. Now these solutions, they can be of various kind. It is hypotonic, hypertonic and isotonic solution. Hypo, hyper and iso. These are now very important terms here, and we need to understand the meaning of each point because they are actually varying in terms of concentration of solute. So if the concentration of solute will change, then definitely the direction in which flow of water will take place, that will also change. Now if it is related to the living cells, then definitely if the flow of water is taking place in or out of the cell, then the shape is going to be affected directly. Because let us assume if there is a cell and water has been moved out, now what will happen? It will get shrink. The opposite, if suppose water moves inside the cell, then what will happen? The cell is going to swell up. So these are the basic things that now we must understand, because the plasma membrane is actually controlling these particular phenomena. Not only the plasma membrane, even the cell wall provides advantage at this particular level. Let us understand the meaning of each kind of solution and of course what happens in such solution. So solution can be hypotonic, hypertonic or isotonic. Hypotonic means a dilute solution. Dilute solution means what? Dilute means in which the concentration of solute is lower. So if solute is low, then definitely the solvent will be high. That means the water level is going to be high. High with respect to what? In comparison to the interior of the cell. Now let us suppose if a cell is placed in a hypotonic solution, then what we actually mean here is that the concentration of solute in the medium or in the solution is low as compared to the solute concentration inside the cell. So now what will happen? The water, it tends to move from outside towards the inside, because here the water content is higher in surrounding condition than that of the interior of the cell. So if outside the water is more, then definitely it tends to move towards inside of the cell, and therefore the cell will get swell up. So such process is known as endoosmosis. And as a result of endoosmosis, what will happen? The cell will get swell up. So in the hypotonic solution, the cell always show the endoosmosis process, and of course they tend to swell up. Oppositely, if it is placed in a hypertonic solution, what we mean by hypertonic? Hyper means high, so the concentration of solute is high. Reversely, the water concentration would be low. Low as compared to the solution, the concentration inside the cell. Now, if the cell is placed in such a medium, then what will happen? As the water is low towards the surrounding and it is high inside the cell cavity, then from inside the water molecule tends to move towards outside. Now this process in which the water molecule tends to move from interior side to the outer side of the cell when placed in a hypertonic solution, then this is known as exo-osmosis. Exo means out, osmosis means movement of water molecules. And if the water has been moved out of the cell, then what will happen to the cell? It will get shrink. So this is what happens when the cells are placed in the hypertonic solution. It always shows exo-osmosis process, and as a result of exo-osmosis, cell will tend to shrink. Further, if the cells are placed in the isotonic medium, then what will happen? But before that, you must understand what we mean by the isotonic solution. Isotonic means the concentration is in equilibrium. It is equal inside as well as outside. So what happens? There is equal movement of water. It will take place, establish a equilibrium. It will establish an equilibrium over there, due to which no change in shape is going to take place. So whenever we need to keep these cells in the laboratory conditions, in vitro condition, that means outside the body, then they have to be kept in a proper medium. And that proper medium which supports the integrity of the cell structure is always a isotonic condition. So we can think of an example here. Let us say, if practically, let us assume, if we keep the raisins, you know what are raisins? The dried grapes. If we keep the dried grapes in a beaker or in a bowl or a glass, in a, let us say, plain water, so what will happen? These raisins, they will tend to swell because interiorly they are having less concentration of the water. So this water will tend to move inside the cells and thus they will get swell up. This you must have observed at your home also. Now this process in which swelling is taking place, that is known as endo-osmosis. The another case in which we are keeping these dried raisins back in a hypertonic condition, then what will happen? They tend to shrink. So this shrinking, it will take place where? In the case of hypertonic, and the process is known as exo-osmosis. Besides, they can be kept in its intact shape if they are placed in the isotonic condition. Now similar process they are also observed in the plant cells. See, in the case of plant cells, we already said there is an additional membrane. There is interior plasma membrane, and this membrane is surrounded by outer non-living coating, and that is known as cell wall. Cell wall is rigid in nature. Fine. So what happens here is, when these cells, plant cells, if they are kept in the hypertonic condition, then what will happen? The water which is present inside the cell, that is plasma membrane, it tends to move out. And as it gets moved out, this interior part is going to get shrink. But there will be no effect over the cell wall because it is non-living as well as it is rigid. So this rigid nature will not change the exterior shape, but definitely interior protoplast is going to get shrink, and thus the cell may die also. Such process in which the water is moving from inside to outside, resulting in the shrinkage of this protoplast, that is known as plasmolysis, and the structure is showing plasmolysed plant cell. Now let us assume if we keep these plant cells in the isotonic condition, then what will happen? We already said there will be an equilibrium established. The amount of water which is moving interior to the cell is always equal to the amount of water which is moving out. So the balance is maintained, and thus this protoplast appears as a flaccid structure. Whereas if suppose we keep it in the hypertonic condition, now what will happen? Sorry, there is a spelling mistake here. In case these are actually, it is placed in the hypotonic condition. It is not hyper, it is hypotonic condition. So hypotonic means dilute condition. So in this case, the water molecules, they tend to move in which direction? It will move from outside to inside. Now as it is taking unidirectionally, it is taking place in a single direction. So what will happen now? The water will tend to move inside, due to which the cell will get swell up. But in this case, it is not going to burst out. Why so? Because there is an outer boundary. This rigid membrane is already present, so it won't allow the protoplast to get burst. Rather, it will appear turgid. So this solid structure is actually it is shown by the plant cells when they are placed in the hypertonic condition. But in the case of animal cells, such support is not provided. So what will happen? If they are placed in the hypertonic, they are simply going to get shrink. If they are placed in the hypotonic condition, but they will swell up, and a time may come when it will get burst. So this is how the movement of material occurs across the plasma membrane. Now let us take an example of a living system also. How this osmosis is taking place in the living system. So the example of this osmosis, it's not diffusion, it is osmosis. Diffusion is actually, along with this, it is also shown here. So it is an example of osmosis as well as the diffusion. Let us explain how it is so. See, this is the plant, and of course the roots are present in the soil. Soil, it is having the water particles. The water layer is always present over there. Besides that, soil also contains the minerals. So what happens here is the water will get mixed up with those elements. Now there are the air spaces also where these are located. Let us see the magnified view of these cells. The roots, they are magnified here. They are having several root hair structures. These root hair structures, they are having a thin membrane here. Now in the surrounding, water is present. This water is mixed up with the elements or the micro and the macronutrients. So the water will move across this particular membrane, because comparatively the level of water inside the root hair is supposed to be less, and that is a true fact. So this water will move from soil into the root hairs, and along with the water it will take up certain molecules inside. Now when these molecules are in high concentration as compared to the interior of the cell, then they will get diffused into the cells, or they will move along with the water and will reach the target site. So this way, this is showing the combined action, the combined phenomena of absorption of water as well as uptake of minerals from the soil by the root hairs. This is all about the movement, and we have taken certain examples. Next we will study about the structure of the plasma membrane and will carry on further. So till then, thank you so much. Have a nice day. Goodbye.